So application of biotechnology in medicine. So a very rapid fire question again. Please tell me, what is diabetes mellitus? How do you diagnose diabetes mellitus? What are the normal blood glucose level? What will be the blood glucose level if we diagnose somebody as a person suffering from diabetes mellitus? What is type 1 diabetes mellitus? What is type 2 diabetes mellitus? What are the key differences between type 1 and type 2 diabetes mellitus? Which is called as insulin dependent, which is called as non-insulin dependent? Which was the first protein for which we came to know about the secondary structure? The name of the scientist was Fred Sanger. He got Nobel Prize for this. So name the protein for which we came to know about the secondary structure, the primary structure. What is the primary structure? So I stand corrected here. Name the protein for which we first came to know about the primary structure. And the primary structure is a linear type of structure which actually tells you the number and sequence of amino acids that is present in a polypeptide or a protein. Again, I told you the name of the scientist associated with Fred Sanger, and which was the first which was the first recombinant therapeutic product for human use. Therapeutic is for treatment. So which was the first approved recombinant therapeutic product for a human disease, so to be used in human beings? So this is an important question. Now, what causes diabetes mellitus? You know, this is insulin deficiency of insulin. Just a few years back, a few decades back, what were the alternatives? Now we will be using recombinant DNA technology, at least in the patients suffering from insulin dependent diabetes mellitus. But what about a few decades back? What was the source of insulin? And what was the problem associated with the use of insulin from these sources in human being? Because there has to be a reason that why are you going for a difficult procedure which is genetic engineering. It is not easy. It is not always successful. So please remember this as a student of biology. Think logically. It is not always that we are trying to do genetic modification and each and everything and it is very simple. We always try something else, and there are some inherent problems in other methods. There are some serious side effects. Then you think of going for recombinant DNA technology. So this please keep this in mind. So again, what were the sources and what was the problem associated with the use of insulin in human beings from these sources? This is an important question, because that is why you will be going for the production of recombinant human insulin. What is it called? What is humulin? So humulin, what is it called? What was the problem? What is a prohormone? How is insulin synthesized in your body? What is a prohormone? How many peptides does it have? What is the sequence of these peptides that is present in the initial transcript? And then which of the peptide is actually discarded and is not used in the final structure of the insulin protein? How many chains does insulin final structure have? What is the number of amino acids in each of the two chains? What links the two chains with each other? All these important questions are there for you. I hope you know the answers. I actually believe that you will be knowing the answers to almost all of them, and now you are feeling confident about it. Yes, we will be able to face the entrance without any difficulty at all. So moving on, then there was a problem. What was the initial problem in the production of recombinant insulin? Which bacteria was used? Were you using plasmid DNA? In this bacteria? What was the initial problem? Which pharmaceutical giant actually got a remedy for this, found a solution for the problem that were being faced in the initial years in the production of recombinant insulin? How this problem was overcome? What is artificial synthesis of gene? Can you synthesize all genes artificially? If not, why? What is the problem here? Is it easy in the case of insulin because because the two chains are actually not having large number of amino acid, and therefore the genes will not be very lengthy, so they will be small genes. And which bacterium, as I told you, was used, and did you use the plasmid of this bacterium, or did you use anything else as a vector? And is there a problem associated with the use of human insulin, and what problem will not be present in the use of human insulin when you compare it with the use of insulin from other sources? So what is this basic thing that we have overcome, and is there any problem associated with human insulin, considering that it is a purified human insulin? And can you anticipate there is a problem? Is it really a problem, or it has been proved that nothing much is to be worried about? So sorry. So these are the questions that you are likely to get from insulin. Very commonly a question is asked. Then the next application in medicine is about gene therapy. What exactly is gene therapy? What is a practical gene therapy? Do you replace a faulty gene with a normal gene, or you provide a normal copy of the gene? So please remember the words. Just listen to me what I am saying. One thing is that you remove the faulty gene and replace it with a normal gene. The second approach can be that you just deliver a normal copy of a gene into the genome of the organism. So which is actually the practical gene therapy? What type of diseases are amenable to gene therapy? Can you think of, say for example, what about sickle cell anemia? What about cystic fibrosis? The diseases that I am mentioning about, is gene therapy a viable option for them in future? And do you think that this gives you an idea that what type of diseases can actually be corrected by gene therapy, at least in future? So this is an important thing. Then what is SCID? Severe combined immunodeficiency. And some cases of severe combined immunodeficiency are caused by the deficiency of an enzyme. What is the name of this enzyme? What exactly is the role of this enzyme in the immune function of the human being? How does it help lymphocytes? You know from Human Health and Disease that adaptive or acquired immunity is a function of lymphocytes. So what exactly is the function of this enzyme? How does it help the lymphocytes in their function? This is a very important thing. Please remember, I have already given you an answer that only certain % of cases suffering from SCID have a deficiency of this enzyme that we are talking about. What would be the problem if a child is born with SCID? What exactly will be the problem? Why the lifespan will not be long? Why there is a problem? There will be very early death. Why do you anticipate that these type of problems will happen? What else other than gene therapy can you try? Is raising the child in a germ-free environment a viable option? If you think it is not, are you aware of the story of David Vetter? So David Vetter, Vetter, David Vetter, who's actually famously known as the bubble boy. You can see the internet and think about it. So I am giving you an option: raising child in a germ-free environment. Is it a viable option? And if yes or if no, then why or why not? And obviously, are there any other issues available? Say for example, psychological issues, keeping a child secluded in a germ-free environment, not allowing the child to intermingle with other children and parents, etc. So will it have a problem in psychological development? What about enzyme replacement therapy? So we know that if somebody is suffering from the deficiency of a hormone, say for example thyroxine, and is having hypothyroidism, a simple treatment can be HRT or hormone replacement therapy. You give thyroxine. This is a very straightforward thing. But what about enzyme replacement therapy? So this enzyme, the deficiency of which we are talking about, is the cause of certain cases, certain % of cases in this SCID. Can we replace the enzyme, and can we do it lifelong? Does it have a role in the treatment of this type of cases of SCID? What about bone marrow transplant? So do you think it is a very, very good option? Do you know that the recipient, the one who is receiving a graft, who is receiving a transplant, can actually reject it? But because this child unfortunately doesn't have immunity, will he or she be able to reject the graft? And if is not able to reject the graft, then bone marrow transplant should be a very, very successful thing. Just try to think on these lines. What is graft versus host disease? What is graft versus host disease, and what is the danger of transplanting something in such patients which actually suffer from severe combined immunodeficiencies? So I have given you 3 options before gene therapy. 1, raising in isolation. 2, enzyme replacement therapy, and 3rd, the bone marrow transplant. Now, then we come to obviously gene therapy. Obviously, now the idea is to deliver a normal copy of a gene. This gene will be making the enzyme, the deficiency of which we are trying to treat. This is now very, very simple. What is the vector used? What is the vector used in the clinical trials for gene therapy for the deficiency of this enzyme, which is responsible for certain percentage of cases of SCID? See, I am not mentioning the name of the enzyme because that may be the question. It's a very simple question in your NEET entrance exam, so I am trying to avoid naming this enzyme again and again. But what was the vector used? What was the vector used? This vector, what was the vector used? And then finally the NCERT says something, which is the cell of the body that you will try to genetically modify. What is the disadvantage of doing it in peripheral lymphocytes or lymphocytes present in the peripheral blood? Would it be a permanent cure? And if you want a permanent cure, what are the cells that you are looking to genetically modify? This is a very, very common question. So this is a common question. Will genetically modifying peripheral lymphocytes work? Will it be a permanent solution? If no, then where are these cells located which may, if genetically modified, may provide a permanent cure for the deficiency of this enzyme? Now, this is what is given in NCERT. I will extend this topic slightly outside NCERT, and just to give you an interesting perspective, many gene therapy trials have resulted in the development of leukemia. So in some cases, in gene therapy for the deficiency of this enzyme, the receivers of gene therapy have actually developed leukemia. What may be the reason for this? What may be the reason that in many instances the gene therapy trials have been put on hold? And we are trying to this question is not for your NEET, but if you have time and you have interest, read about adeno-associated virus, AAV, and how can in future it help us overcoming the problem for which we have actually halted many gene therapy trials over the world. And please remember what type of diseases would be amenable to gene therapy. I hope all these questions are now absolutely clear. These are the possible questions that can be asked from this gene therapy section of applications of biotechnology in medicine. The final application in medicine would be molecular diagnosis. In the previous chapter, we have already talked about PCR. So we know what is PCR, polymerase chain reaction. You know the step of polymerase chain reaction. You know about Taq polymerase. You know about PFU polymerase. You know about Kary-Mullis. You know what are the different temperature ranges required for every step of polymerase chain reaction, and what exactly is it used for. Now the diagnosis part. How can PCR help in early diagnosis of the disease? How can it help you differentiate between diseases that mimic one another? They may produce similar clinical sign and symptom, but their treatment may actually be widely different. How does PCR help you differentiating these closely mimicking clinical conditions? And similarly, how can PCR be actually used in actually understanding the risk of development of a cancer in a person? How can you calculate with the help of PCR that there is a certain risk of development of cancer in such person? So this is about PCR. The second thing it talks about is ELISA. First is the full form. What is the full form of ELISA? Second, what is the basic principle on which ELISA functions. What do you detect in a given sample? What do you detect in a given sample? And is ELISA a screening test or a confirmatory test in most of the cases? So this is a very important thing to understand. Is ELISA a very good screening test, or in most cases is it a confirmatory test? What is the drawback of ELISA test, and what exactly is a sandwich ELISA? What is capture antibody? All these things can be asked. Again, in a sample you are detecting something to diagnose a disease with the help of ELISA. What are these some things? Do you know what are the type of antibodies produced during a primary response? If you remember human health and disease, IgM and IgG, which one is primary, which one is secondary, and what would be the diagnostic importance of ELISA if we come to know that a person has IgG positive or IgM positive for a certain antigen? So these are the questions that can be asked in molecular diagnosis, the section of your NCERT